So, hello everyone. So, today we are going to look into the comprehensive analysis of TNF gene. So, our first task would be the download a biological sequence from NCBI. So, let's get into the task one. So, before moving on into practical session, we just want to give a brief introduction about how it works, what you are going to learn, how to download the file from which database. So in this task, the first thing is we have to download our biological sequence, that is our genomic sequence, from which database? That is NCBI database, that is National Center for Biotechnology Information Database. So it is very important that this allows us to access particular genomic database, and we can also manipulate the sequence in the various bioinformatic analysis. So here, many biological sequence, as you see, there is some 3 code string letters that can represent whether the amino acid is nucleotide or nucleoside that can be denoted as a DNA or RNA or proteins. So it is like a genetic information. So it is used in many areas in biology, medicine, including our gene expression, evolution, and some diagnostic of diseases. The specific amino acids are A denotes adenine, T denotes thymine, C cytosine, and G thymine. Okay. So the first step what we are going to do is, so we are going to access the NCBI database. For that, we have to go to the home page of NCBI. So it looks like this. As image I have mentioned, it looks like this. And you can go to directly to Google and type ncbi.nlm.nih.z, or you can simply type NCBI. So you will reach to this database like this homepage. Next, particular sequence. So the type and the name of the sequence is TP53, tumor suppressor gene. So you just enter which you just change the gene and enter the name of the gene and press enter. As it is in the search result, you will have a sequence records that can match your query. What is the query you have entered? That is TP53. So our next step is we have to choose the sequence. So there are many sequence queries, so we have to choose a specific sequence record page. So where you can find human beings, okay? So you have to select according to the which animal you are going to get the sequence. So you have to select Homo sapiens, that is our humans. So you will find different information of the particular gene. So you just gradually scroll down. So you can see the genomic region section. Then click on the FASTA sequence. As you can see the send to, you can click on that send to, and you will find this FASTA page. Okay, when you see it, and you click on to send to. Okay. So this will take you to the reference sequence page. Gradually, you can view the full sequence by scrolling on down the page when you click on the FASTA sequence. After clicking on to the FASTA sequence, what you are going to do is, like you are going to send it to, how you are going to download this FASTA file. That is our next step. So we are going to download the sequence to take send to, click on the button that is complete record unit. So select file in the drop menu, choose the format, which format you need to download the sequence. So you can enter the format as FASTA. You can select the choose destination as a file, and you can choose the complete record. And Click on create file. Now what we'll be getting is before we end, so we have to download and analyze these biological sequence. Is very important because it allows us to manipulate particular sequence in various bioinformatic analysis. So many of the researchers or bioinformaticians or analysts might need to download and analyze the sequence of the gene from different individuals to identify mutations that are being associated with some certain disease or trend. So there are many databases with respect to genomics. Not only NCBI, you have other databases also where you can download the genomics or gene sequence. So now let's move on to the hands-on practical session. Now we have reached to the Google. So now what we are going to do is we are going to type in C I. Okay, just click enter. So it is loading. So now click on to the National Center of Biotechnological Information. Just click on to the first icon. Now you have reached your homepage of your NCBI. So we have to select G. Okay. Next we have to type TNF Gene. Okay. Give search. Then you can find a number of sequence queries. The query which you have entered specific to that, you can reach n number of genes with respect to mouse, with respect to rat, and we also have humans. That is how human Homo sapiens. So we are going to work with it. So what you are going to do is just click on TNF gene. Then you can see the official symbol of the gene name and official name, that is human necrosis factor, and the primary sources, gene type which included, and the reference status and which organism that it has been studied, and the summary and the annotation information and the expression system. So you can see as you scroll down, so gradually. So here what we are going to do, here you can look at the name for faster. So you have to click faster. and it will direct to the FASTA sequence page. Now you can see the reference. This is your reference sequence ID, and this is Homo sapien and the chromosome, the location. That is our primary assembly. So here you can see the FASTA sequence. Now what we are going to do is, the next step, we are going to download this sequence. So just go, see, just click on to complete record and choose the destination, that is file, and which format we need. We need FASTA format, and we have to click the create file, right? So once we downloaded the file, the next step is we are going to see the sequence. We are going to view and we are going to edit using BioEdit software. Okay. So next what we are going to do is, next we are going to move to the BioEdit software. So this is called BioEdit sequence alignment editor, where you can able to see your sequence. So what I'm going to do, I'm going to load sequence which I have downloaded. Now it will direct me to the download page, and you can upload the FASTA file to it. Now we have to see, here we have to change, if you always you have to look up on the file of the type. See, if you look for the gene pan file, it won't be available. So if you look for a FASTA file, click here and you will see the visible file. So I have already worked with this sequence. So I have downloaded multiple times. So it is, so I'm taking the download a file, that is our sequence. So whichever sequence you need, you can just download it and you can just view it and edit it using this BioEdit software. So as you can see, this is our sequence that we have loaded. I hope you like this task one. So please do follow and like my channel and complete your task one and let us know in the comment section. Please like and share it so others can learn it too along with you. And thank you. Hope we will see in the next task too.